Neisseria gonorrhoeae, Wikipedia article audio. Neisseria gonorrhoeae, also known as gonococcus, or gonococcus, is a species of gram-negative diplococci bacteria isolated by Albert Neisseria in 1879. It causes the sexually transmitted genitourinary infection gonorrhea as well as other forms of gonococcal disease including disseminated gonococcemia, septic arthritis, and gonococcal ophthalmia neonatorum. Microbiology. Culture and identification. Surface proteins. Antigenic variation. Survival of gonococci. Genome. Horizontal gene transfer. Disease. Transmission. Prevention. Treatment. Antibiotic resistance. History. Name origin. Discovery. It is oxidase positive, aerobic, and it survives within neutrophils. To culture, it requires carbon dioxide supplementation and enriched agar with various antimicrobials. It exhibits antigenic variation through recombination of its pili and surface proteins that interact with the immune system. Sexual transmission is possible through vaginal, anal, or oral sex. Sexual transmission may be prevented through the use of barrier protection. Perinatal transmission may occur during childbirth, and may be prevented by antibiotic treatment of the mother before birth and the application of antibiotic eye gel on the eyes of the newborn. After an episode of gonococcal infection, infected persons do not develop immunity to future infections. Reinfection is possible due to N. gonorrhoeae's ability to evade the immune system by varying its surface proteins. N. gonorrhoeae can cause infection of the genitals, throat, and eyes. Asymptomatic infection is common in males and females. Untreated infection may spread to the rest of the body, especially the joints. Untreated infection in women may cause pelvic inflammatory disease and possible infertility due to the resulting scarring. Diagnosis is through culture, gram stain, or PCR of a urine sample, urethral swab, or cervical swab. Chlamydia CO testing and testing for other STIs is recommended due to high rates of co-infection. Neisseria species are fastidious gram-negative cocci that require nutrient supplementation to grow in laboratory cultures. Neisseria are facultatively intracellular and typically appear in pairs, resembling the shape of coffee beans. Neisseria is non-spore-forming, non-motile and an obligate aerobe. Of the 11 species of Neisseria that colonize humans, only 2 are pathogens. N. gonorrhoeae is the causative agent of gonorrhea and N. meningitidis is one cause of bacterial meningitis. Neisseria gonorrhoeae is usually isolated on Thayer-Martin agar-agar in an environment enriched with 3 to 7% carbon dioxide. Thayer-Martin agar is a chocolate agar plate containing nutrients and antimicrobials. This agar preparation facilitates the growth of Neisseria species while inhibiting the growth of contaminating bacteria and fungi. Martin Lewis and New York City are other types of selective chocolate agar commonly used for Neisseria growth. N. gonorrhoeae is oxidase positive and catalase positive. When incubated with the carbohydrates lactose, maltose, sucrose, and glucose, N. gonorrhoeae will oxidize only the glucose. On its surface, N. gonorrhoeae bears hair-like pili, surface proteins with various functions, and sugars called lipooligosaccharides. The pili mediate adherence, movement, and DNA exchange. The OPA proteins interact with the immune system, as do the porins. Lipooligosaccharide is an endotoxin that provokes an immune response. All are antigenic and all exhibit antigenic variation. The pili exhibit the most variation. The pili, OPA proteins, porins, and even the LOS have mechanisms to inhibit the immune response, making asymptomatic infection possible. Dynamic polymeric protein filaments called type IV pili allow N-gonorrhoeae to adhere to and move along surfaces. To enter the host, the bacteria uses the pili to adhere to and penetrate mucosal surfaces. The pili are a necessary virulence factor for N-gonorrhoeae. Without them, the bacteria is unable to cause infection. To move, the bacteria uses the type IV pili like a grappling hook, extending and retracting it for movement. Pili extend and attach to a substrate that signals the pilus to retract, dragging the cell forward. The resulting movement is referred to as T-witching motility. N-gonorrhoeae is able to pull 100,000 times its own weight, and it has been claimed that the pili used to do so are the strongest biological motor known to date, exerting 1 nanonewton. A set of ADPase proteins power the pulling actions of the type IV pilus, pil F and pil T. The adhesive functions of the gonococcal pilus plays a role in microcolony aggregation and biofilm formation. Surface proteins called OPA proteins can be used to bind to receptors on immune cells and prevent an immune response. There are at least 12 OPA proteins, and the many permutations of surface proteins make it more difficult for immune cells to recognize N-gonorrhoeae and mount a defense. Lipooligosaccharide is a low-weight version of lipopolysaccharide present on the surfaces of most other gram-negative bacteria. It is a sugar side chain attached to lipid A in the outer membrane coating the cell wall of the bacteria. The root oligo refers to the fact that it is a few sugars shorter than the typical lipopolysaccharide. As an endotoxin, LOS provokes inflammation. The shedding of LOS by the bacteria is responsible for local injury in, for example, pelvic inflammatory disease. Although its main function is as an endotoxin, LOS may disguise itself with host sialic acid and block initiation of the complement cascade. N-gonorrhoeae evades the immune system through a process called antigenic variation, in which the N-gonorrhoeae bacterium is able to recombine its genes and alter the antigenic determinants such as the OPA proteins and type IV pili that adorn its surface. It is able to vary the composition of its pili, OPA proteins, and lipooligosaccharides. Of these, the pili exhibit the most antigenic variation due to chromosomal rearrangement. Every time the bacteria replicate, they may switch multiple OPA proteins on or off through slip-strand mispairing. That is, the bacteria introduces frame-shift mutations that bring genes in or out of frame. The result is that different OPA genes are translated every time. In addition to the ability to rearrange the genes it already has, it is also naturally competent to acquire new DNA, via its type IV pilus, specifically proteins pil Q and pil T. These processes allow N-gonorrhoeae to acquire/spread new genes, disguise itself with different surface proteins, and prevent the development of immunological memory inability, which has led to antibiotic resistance and has made vaccine development difficult. After gonococci invade and transcytose the host epithelial cells, they land in the submucosa, where neutrophils promptly consume them. The pili and OPA proteins on the surface may interfere with phagocytosis, but most gonococci end up in neutrophils. The exudates from infected individuals contain many neutrophils with ingested gonorrhoeae. Neutrophils release an oxidative burst of reactive oxygen species in their phagosomes in order to kill the gonorrhoeae. However, a significant fraction of the gonorrhoeae can resist killing through the action of their catalase, which breaks down reactive oxygen species and are able to reproduce within the neutrophil phagosomes. Stoll and Seifert showed that the bacterial RECA protein, which mediates repair of DNA damage, plays an important role in gonococcal survival. Micode et al. Have suggested that N-gonorrhoeae may replace DNA damaged in neutrophil phagosomes with DNA from neighboring gonorrhoeae. The process in which recipient gonorrhoeae integrate DNA from neighboring gonorrhoeae into their genome is called transformation. The genomes of several strains of N-gonorrhoeae have been sequenced. Most of them are about 2.1 MB in size and encode 2,100 to 2,600 proteins. For instance, strain NCCP11945 consists of one circular chromosome encoding 2,662 predicted ORFs and one plasmid encoding 12 predicted ORFs. The estimated coding density over the entire genome is 87%, and the average GNC content is 52.4%, values that are similar to those of strain FA1090. The NCCP11945 genome encodes 54 tRNAs and 4 copies of 16S23S5S rRNA operons. In 2011, researchers at Northwestern University found evidence of a human DNA fragment in a Neisseria gonorrhoeae genome, the first example of horizontal gene transfer from humans to a bacterial pathogen. Symptoms of infection with N-gonorrhoeae differ, depending on the site of infection. Note also that many infected men are asymptomatic and most infected females are asymptomatic. In symptomatic men, the primary symptom of genitourinary infection is urethritis, burning with urination, increased urge to urinate, and a pus-like discharge from the penis. The discharge may be foul-smelling. If untreated, scarring of the urethra may result in difficulty urinating. Infection may spread from the urethra in the penis to nearby structures including the testicles, or to the prostate. Men who have had a gonorrhea infection have a significantly increased risk of having prostate cancer. In symptomatic women, the primary symptoms of genitourinary infection are increased vaginal discharge, burning with urination, increased urge to urinate, pain with intercourse, or menstrual abnormalities. Pelvic inflammatory disease results if N-gonorrhoeae ascends into the pelvic peritoneum. The resulting inflammation and scarring of the fallopian tubes can lead to infertility and increased risk of ectopic pregnancy. Pelvic inflammatory disease develops in 10 to 20% of the females infected with N-gonorrhoeae. N-gonorrhoeae may cause infection of the throat through oral sex. Infected persons may experience sore throat and swollen lymph nodes, or may be asymptomatic. N-gonorrhoeae may cause infection of the anus/rectum through anal sex. Infected persons may experience blood or a pus-like discharge, or may be asymptomatic. In perinatal infection, the primary manifestation is infection of the eye when the newborn is exposed to N-gonorrhoeae in the birth canal. The eye infection can lead to corneal scarring or perforation, ultimately resulting in blindness. If the newborn is exposed during birth, conjunctivitis occurs within 2 to 5 days after birth and is severe. Gonococcal ophthalmia neonatorum, once common in newborns, is prevented by the application of erythromycin gel to the eyes of babies at birth as a public health measure. Silver nitrate is no longer used in the United States. Disseminated gonorrhoeae infections can occur in which gonococci enter the bloodstream, often spreading to the joints and causing a rash. Dermatitis arthritis syndrome presents with joint pain, tendon inflammation, and painless non-pruritic dermatitis. Disseminated infection and PID tend to begin after menses due to reflux during menses, facilitating spread. Rarely, disseminated infection may cause infection of the meninges of the brain and spinal cord or infection of the heart valves. Gonorrhoeae may be transmitted through vaginal, oral, or anal sex. Non-sexual transmission is unlikely in adult infection. It can also be transmitted to the newborn during passage through the birth canal if the mother has untreated genitourinary infection. Given the high rate of asymptomatic infection, all pregnant women should be tested for gonorrhea infection. Traditionally, the bacteria was thought to move attached to spermatozoon, but this hypothesis did not explain female-to-male transmission of the disease. A recent study suggests that rather than surf on wiggling sperm, gonorrhoeae bacteria uses hair-like structures called pili to anchor onto proteins in the sperm and move through coital liquid. Transmission can be reduced by using latex barriers during sex and by limiting sexual partners. Condoms and dental dams should be used during oral and anal sex as well. Spermicides, vaginal foams, and douches are not effective for prevention of transmission. Ceftriaxone is the first-line treatment. Because CO infection with chlamydia is so common, people are often treated for both by combining ceftriaxone with azithromycin or doxycycline. Azithromycin is preferred for additional coverage of resistant gonorrhea. Sexual partners should also be notified, tested, and treated. Antibiotic resistance in gonorrhea has been noted beginning in the 1940s. Gonorrhea was treated with penicillin, but doses had to be progressively increased in order to remain effective. By the 1970s, penicillin and tetracycline-resistant gonorrhea emerged in the Pacific Basin. These resistant strains then spread to Hawaii, California, the rest of the United States, and Europe. Fluoroquinolones were the next line of defense, but soon resistance to this antibiotic emerged as well. Since 2007, standard treatment has been third-generation cephalosporins, such as ceftriaxone, which are considered to be our last line of defense. Recently, a high-level ceftriaxone-resistant strain of gonorrhea called H041 was discovered in Japan. Lab tests found it to be resistant to high concentrations of ceftriaxone, as well as most of the other antibiotics tested. Within gonorrhoeae, there are genes that confer resistance to every single antibiotic used to cure gonorrhea, but thus far they do not coexist within a single gonococcus. However, because of gonorrhoea's high affinity for horizontal gene transfer, antibiotic-resistant gonorrhea is seen as an emerging public health threat. Neisseria gonorrhoeae is named for Albert Neisseria, who isolated it as the causative agent of the disease gonorrhea in 1878. Galen coined the term gonorrhea from the Greek gonos, which means seed, and rho, which means flow. Thus gonorrhea means flow of seed, a description referring to the white penile discharge, assumed to be semen, seen in male infection. In 1878, Albert Neisseria isolated and visualized gonorrhoeae diplococci in samples of pus from 35 men and women with the classic symptoms of genitourinary infection with gonorrhea, 2 of whom also had infections of the eyes. In 1882, Leistikow and Loeffler were able to grow the organism in culture. Then in 1883 Max Bockhardt proved conclusively that the bacterium isolated by Albert Neisseria was the causative agent of the disease known as gonorrhea by inoculating the penis of a healthy man with the bacteria. The man developed the classic symptoms of gonorrhea days after satisfying the last of Koch's postulates. Until this point, researchers debated whether syphilis and gonorrhea were manifestations of the same disease or two distinct entities. One such 18th-century researcher, John Hunter, tried to settle the debate in 1767 by inoculating a man with pus from a patient with gonorrhea. He erroneously concluded that both diseases were indeed the same when the man developed the copper-colored rash classic syphilis, respectively. Although many sources repeat that Hunter inoculated himself, others have argued that it was in fact another man. After Hunter's experiment, other scientists sought to disprove his conclusions by inoculating other male physicians, medical students, and incarcerated men with gonorrheal pus, who all developed the burning and discharge of gonorrhea. One researcher, Rickard, took the initiative to perform 667 inoculations of gonorrheal pus on patients of a mental hospital, with 0 cases of syphilis. Notably, it was not until the advent of penicillin in the 1940s when effective treatments for gonorrhea were available.